I am Nicole LeDouarin. I am an honorary professor at the Collège de France in Paris. I'm going to talk to you today about the neural crest, because I am an embryologist, and the role of this structure in the development and in the evolution of vertebrates. The neural crest is a transitory structure of the vertebrate embryo, and it is also a pluripotent structure. It has been discovered by the German histologist Wilhelm His in 1868 in the avian embryo. By looking at the living embryo under the microscope, Wilhelm His noticed on the dorsal aspect of the neural tube a strand of cells lying there. And after a while, he saw that the cells which constituted this structure were moving away from the neural primordium and were aggregating laterally to form the the dorsal root ganglia. This is why the first name that he attributed to this structure was the ganglionic crest. This observation aroused the interest of many embryologists at that time, and they worked essentially on the neural crest of lower vertebrates, amphibians and fish. And the work which was done during this period, the first half of the 20th century, was summarized in a well-acknowledged monograph written by Sven Orstadius in 1950. In the late 1960s, when I started to be interested in the neural crest, very little was known about the role of this structure in the embryogenesis of higher vertebrates. And the reason for this is because in amniotes, vertebrates, the number of cells in the embryo becomes high, and the cells which are migrating are rapidly undistinguishable from the cells of the tissues through which they move. This problem was overcome after I devised a cell marking technique which can be applied in the avian embryo. My talk today will be divided into three parts. The first part will be the description and the use of the quail cheek marker system, and it's used to study the ontogeny of the neural crest. The second part will deal with the role of the neural crest in the development of the head of vertebrates and also in the evolution of vertebrates. The third part will concern the molecular control of the contribution of the neural crest to craniofacial and brain development. The cell marking technique that I devised consists in constructing chimeras in ovo between two different species of birds, the chick, Gallus, which was actually a common material for embryologists at that time, and another bird, the Coturnix japonica, that means the quail that you see on this side of the slide. You see that the two birds at birth have different size, and they also have a different duration of incubation period, 21 days for the chick and 17 days for the quail. However, the size of the embryos during the first half of incubation period, when most of the important events in development take place, is about the same in the two species. The technique that I devised is based on an observation that I made about the structure of the nucleus in these two species. I noticed that in all embryonic and adult cell types of the quail, there is a particularity in the nucleus. And this particularity consists in the fact that the heterochromatic DNA is concentrated in the center of the nucleus in a large mass, which can be stained by a staining procedure which is specific for DNA, the Fulgen procedure. And this heterochromatic DNA is always associated with the nucleolar RNP, making the nucleolus, which is unusual, Fulgen positive. This does not exist in the chicks that you see in this part of the slide, where, like in man, like in mouse, and like in most animal species, the heterochromatin is evenly dispersed in the nucleoplasm during the interphase. I had the idea of using this particularity of the quail nucleus, which is a stable marker, to devise a cell marking technique to follow the migration, the movements of the cells during embryogenesis, and also to follow their fate. The idea was to remove from the chick embryo, for example, the part of the embryo which is the fate of which is under scrutiny, and to replace it by the equivalent region coming from a quail embryo at exactly the same developmental stage. These chimeras develop normally, and the quail cells which have been implanted can be found anytime after grafting, thanks to the particularity of the quail nucleus of the nucleus of the quail by using the Fulgen staining. Now it is possible to. It has been possible from 1975 to prepare monoclonal antibodies, which are specific for antigens which are present only on quail cells and not on chick cells. One of those monoclonal antibodies, which is called the QCPN for quail non-chick perinuclear antigen, has been prepared in the University of Michigan by Carlson, and it is very much used to analyze these chimeric quail chick embryos. This technique can be combined with the use of molecular probes, and transgenesis is also possible by using electroporation to introduce nucleic acid into definite regions of the chick embryo. One of the most interesting application of the quail chick marker system was to study cells which have the property of migrating inside the embryo during embryogenesis. This is the case of the neural crest and also of hemopoietic cells. And in my laboratory during the past years, those subjects, the neural crest and also the development of hemopoietic organs, have been the main subjects that were under the study. The neural crest in the avian embryo is derived, like in the other embryos, from the lateral border of the neural primordium, which are called the neural folds. Closure of the neural tube starts at the level of the mesencephalon, that you can see here, and then proceeds rostrally and caudally along the future spinal cord. In the spinal cord, the neural folds are less conspicuous than at the head level. However, the process of closure of the neural tube proceeds in the same way. After fusion of the neural folds, the neural crest cells, which are here, have lost their epithelial arrangement. And they are rapidly going to migrate into the embryo along definite pathways at precise period of time, and they will settle in elected points in the embryo where they will differentiate into a large variety of derivatives. It has been shown rather in the recent years that at this stage the neural crest cells can be distinguished from the cells of the neural primordium, the cells of the neural tube, and also from the cells of the superficial ectoderm, by the fact that they expressed a certain number of genes, and particularly of transcription factors, which are listed here, for example, slug, genes of the SOX family, SOX8, 9, and 10, FOXD3, and also PAX3. And they also carry glycoprotein, which has an epitope, a glycosylated epitope, which is recognized by the monoclonal antibody HNK1. The principle of the experiments that we have used in order to study the neural crest cells and their fate, their migration, is as follows. We take a chick embryo as a host, and we removed at the definite region of the neural axis, here, we remove the neural tube which has just closed, but from which the neural crest cells have not yet started to migrate. Then we take a quail embryo, which will be the donor embryo, and in which we isolate the neural tube from exactly the same level by subjected this region to trypsinization in order that the neural tube, which is going to be grafted, is not contaminated by other cells of different origin. And the neural tube is then implanted into the chick host at the site where the excision of its own neural tube has taken place. And the next slide shows the real experiments in which the chick embryo has been prepared for the graft by removing of the tube. You see here the notochord, and on each side the somites. And this is the neural tube of the quail, which is going to be implanted. And you see on the top the neural crest cells. And about 12 hours after implantation, you see that the neural tube of the quail has developed normally. It has been covered by the healing host ectoderm. And in this slide, it is possible to distinguish the cells coming from the neural crest of the quail, which has neural tube which has been implanted, and which are migrating within the cells of the chick, from which they are easy to distinguish because of the structure of their nucleus. These chimeras develop normally, and in this case, the host embryo was of the white Leghorn strain. That means it was completely devoid of pigment. And you see that the only sign that this bird is a chimera is the transverse stripe of quail-like pigmented feathers. These feathers, of course, belong to the chick, but they have been colonized by the precursors of melanoblasts, which arise from the quail neural tube which has been implanted into the chick. These birds can hatch. You can see here quail chick chimera, quail chick neural chimera, which is 3 months old. It can walk, it can fly, it can compete for food, Therefore, it has a normal behavior, which means that the synapses between the quail neurons and cheek neurons, as well as the synapses between the quail neurons and the cheek muscles, are absolutely functional, normal. And this means that this chimeric model is quite reliable to study the development of the nervous system and the development of the neural crest. This technique, which was these types of graphs, which were carried out systematically along the whole neural axis by Marie Métayer, Christian Lelièvre, and myself in the 70s, has led us to complete the list of the neural crest derivatives. The derivatives of the neural crest are numerous and diversified. They include the neurons of the peripheral nervous system. That means the sensory neurons, the sympathetic and parasympathetic neurons, and also all the nervous system which is present inside the enteric, the enteric system, that means the digestive tract. The neural crest also provides this peripheral nervous system with glial cells. That means with the satellite cells which accompany the neurons in the ganglia, peripheral ganglia, and the axons in the peripheral nerves. That means that the neural crest is at the origin of the Schwann cells. It is also at the origin of the pigment cells of the body, all the pigment cells except those of the pigmented retina. It is at the origin of endocrine cells, the adrenal medullary cells, which produce epinephrine and norepinephrine, and also the so-called C cells of the thyroid gland, that means the calcitonin-producing cells of the thyroid gland. In addition to all these cell types, the neural crest is also, and more surprisingly, at the origin of mesenchymal cells. And they were called by the person who discovered them, Julia Platt, at the end of the 19th century, mesectoderm, to distinguish these mesenchymal cells from the mesenchymal cells of the mesoderm, which are the mesenchymal cells which are all over the body, except at the head level, where the mesenchymal cells are derived from the neural crest and are called mesectoderm. The cell types which arise from mesectoderm are cartilage, bones. They form the connective tissue of the dermis, connective tissue associated with the striated muscles, which are at the head level, like the masticatory muscles, for example. They form the vascular smooth muscle cells and also the pericytes, and they form also adipocytes, for example, adipocytes in the dermis. In addition to this list of derivatives that was derived from, or for many of these cell types from our studies and studies which were done before, we were also able to distinguish from which level of the neural axis all these different cell types arise. And what is important is, as I said, that the mesectoderm rises only from the neural tube which is at the head level. So that means is the cephalic neural crest that you see here. The trunk neural crest, as we have seen, does not give rise to mesenchymal cell types. The cells which are going to become pigment cells arise from the whole length of the neural axis. And as far as the ganglia of the peripheral nervous system are concerned, their origin along the neural tube is regionalized. And I'm going to give you an example of this. As you can see here, we have indicated the gut on the left, and the gut is interesting for us because it is invaded by neural crest cells, which form two plexuses in the gut, the myenteric plexus and the submucosal plexus. And the origin of the cells of this plexus are located anteriorly from the level of somite 1 to the level of somite 7. The cells migrate massively from this region, go to the pharynx, and when they are in the wall of the gut in the pharynx, they migrate all along the gut down to the cloaca. There is a secondary supply of cells to form these gut plexuses, which arise from the lumbosacral region of the neural tube, and which concerns only the post-umbilical gut. There is another region which is of interest. This is a region which gives rise to the sympathetic ganglia, and you see that the superior cervical ganglion arise from cells which are at the level of somite 6. The other region which is of interest concerns the region from which the adrenal medulla arises. And it is from this level, which is from somite 18 to somite 21, that means about the level of the wing in the birds. And I'm going to show you experiments which are the result of first of the replacement of the neural tube at the level of somite 18 to 24. And this is a section of the suprarenal gland of such a chimera. You see on the left that there are strands of cells which are fluorescent. And this fluorescence is a result of the so-called Falk and Hillarp technique, which allows the catecholamines to be evidenced inside the cells. So those cells are cells which are those which produce adrenaline and noradrenaline. And the same section was subjected after to the Fulgen reaction. And you see it on the right side. And if you superimpose the two sections, you see that the cells which contain catecholamines also contain the nucleolus of the quail. And the cells which are between these strands of cells, which are catecholaminergic, there are cells which have the chick nucleus. And the cells which have the chick nucleus are derived from the chick embryo host, and they are the cells which produce the corticosteroid of the suprarenal gland. Now, let us see the replacement of the vagal region of the neural crest, that means the anterior region from somite 1 to somite 7. I call it vagal region because this is a region from which the roots of the vagus nerve, which innervates the gut and forms the preganglionic fibers, which innervates the plexuses of the gut, arise. And this is a region from which the neural crest cells, which are going to invade the gut, arise also. And I'm going to show you an experiment which was done by Alan Burns in the laboratory, and the same experiment were done by myself and also by Marie Métayer quite routinely. You see the chick embryo on the left, in which the neural tube has been removed from somite 1 to somite 7. And what you see between the somites is the notochord. You see on the right the experiments, which consist in taking a quail embryo exactly at the same stage, removing the same level of the neural tube, and the neural tube, which is red there, is implanted in the chick after. And you see the result of the experiment, which was in which the quail cells were evidenced by the antibody, QCPN antibody. You see how clean the experiment is and how well the neural tube of which has been grafted is incorporated into the host. And you see here the migration of neural crest cells, which reach the ventral region. And you see finally a part of the gut, the embryonic gut, of course, in which the quail cells are stained with the QCPM antibody. And you see that the submucosal plexus, which is intern, and the myenteric plexus are being formed on each side of the circular muscle layer.